The first homework question was for the cross-section of the cuticle layer, the outermost region of a nematode. I've got two examples here for you that show this pretty nicely, and there's only a couple things I would add. So the outermost layer here is lipid. It's only about 10 microns, so pretty thin. And then underneath it is a region of collagen. So I probably would have added a really thin line here and pointed out that that's the epicuticle of lipid. The lipid it tends to be tough, pretty waxy lipids for these kinds of outermost layers in a lot of animals. And when we talk about waxy lipids, we're usually talking about long carbon chains, which each of these dots being a carbon. So carbon, 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 carbon. And with just a few branches or things like that, they tend to pack really well together. And they form barriers that are impervious to water. And they also are really hard for microorganisms to get through. So that's this outermost layer. And that's about as thick as a cell layer in a nematode, about 10 microns. Then there's collagen here. There's more collagen support. It's not quite clear why that's there, at least not clear to me. And then these helical fibers that are fibers of collagen oriented in different directions. And some of them are oriented in helices around the whole animal. So we'll come back to that a little bit later. Then the other thing I would add is in the what they call the HYP layer here, I would add cells so that you could actually see cells here. And then underneath it, of course, this is shown in this homework. There's the basement membrane, which, by the way, is also called basal lamina. They aren't actually quite the same. One of them is just a component of the other, but they're used interchangeably so often that people often say basement membrane and basal lamina and mean the same thing, and that's how I use them in this course. Here's another version that just shows the same thing. Again, I would have put in the cell layers here for the epithelial tissue. Basement membrane here would only be about 100 nanometers, whereas the epithelial layer likely would be 10 or 20 nanometers, likely a single cell layer for the cells of the epithelial layer. For 2, this was a really beautiful example, so I just had to use it. Much better than I could draw. I liked a number of things in it. It's really nicely done, showing the gut. So here's the gut, the muscular pharynx for crushing food, and then area where things are being absorbed. Here are the longitudinal muscle fibers, showing them long and slender here. See the epicuticle around the outside, the pseudocoelom, the space in here. An advantage of having a coelom is that when muscles contract and the body bends, that the organs don't have to move with the whole body. They can slide back and forth. When you twist your body around, your heart and your liver and things can stay more or less where they are. You don't change the pressure on your intestine just by moving a lot. If you don't have a coelom, when you move your body, that rearranges all your internal organs. So things like a flatworm with no coelom, when they bend their body, they bend everything, whereas pseudocoeloms don't. And then pseudocoeloms and coeloms also can be used as a hydrostatic skeleton, which we'll come back to. So there's the pseudoskeleton. The helical fibers are wrapped around the body like this. So we go here, and then here, and then here. So they're disappearing behind the body, coming out again, disappearing behind the body, coming out again, disappearing behind the body. So they're in this outermost layer going wrapped around like this. What that does for a nematode is that every time it contracts its longitudinal muscles on one side, it bends in that direction. Wherever it contracts its longitudinal muscles, it bends in that direction. As soon as it relaxes, then elastic recoil will bring it back. There's a point of confusion here that we're going to come back to about how opposing muscles work. But something always has to bring them back. A muscle can't simply relax and go back to its original length. It has to either have an opposing muscle In this case, the muscles on the other side could do it, or it needs elastic recoil, and that's mainly what it uses. Nematodes use elastic recoil. When they bend, these collagen fibers resist that bending. So they bend under the muscle contraction, but as soon as the muscle relaxes again, elastic recoil brings it back. And what that means is that a nematode can have only longitudinal muscle and still be able to move. It doesn't move like an earthworm. It can't do body extension, so it doesn't have circular muscle, so it can't lengthen the body and get thinner like an earthworm can. All it does is these thrashing back and forth movements, but it makes it very simple to control. The next one was heart function. I gave a couple examples. This person showed the muscles contracting, forcing, so creating pressure here. That pressure causes back pressure against these two valves, so they were open, but with the back pressure, they're now closed. This pressure here forces these valves that had been closed, forces them open. So here are the arrows showing these valves going open, and out goes the blood. To refill, you either need blood coming in under pressure, that's what your heart does, or in some cases there is elastic recoil. So if there's something here, fibers there or fibers there, that can have elastic recoil, then when the muscle relaxes, it goes back to its original shape. Or new blood or hemolymph coming in under pressure, that pressure will open these valves. Or if this is elastic recoil, you get a partial vacuum that when the pressure is lowered, when this is relaxing, back pressure from the blood that was just pumped out pushes these valves closed, because now this is higher pressure from the blood just pumped in, this is lower pressure. The valves get closed so the blood can't go back, but the lower pressure in or the higher pressure outside forces these valves open and blood refills, and then we contract again. This person showed it with an atrium and a ventricle, showing the atrium filled with blood here and then contracting. So when it's filling with blood, blood is coming in here. These valves should actually be closed. Blood is coming in here. When the atrium contracts, that forces these valves closed because of the back pressure, and out goes the blood. Then when the ventricle, much thicker walled, so atria can be very thin because all they have to do is move blood from here to here. The ventricle, much thicker walled, more muscle, because they have to pump blood through the whole rest of the body. So when the ventricle contracts, that as it starts to contract, that creates back pressure here, so these valves closed. These were closed when blood was filling, but when the ventricle is contracting, it forces out the blood, or in an arthropod, hemolymph, out, and then at the same time you can have atrial filling, back pressure closing here, and either negative pressure in here or maybe pressure coming from the hemolymph returning refills the atrium. This also shows the heart with colors to show the two stages of contraction. So the contraction when blood is being pumped out, and then refilling when blood is coming back in. And here is a arthropod open circulatory system where you have an atrium, which is pumping blood into a ventricle, thicker walled because it's got a lot further to go. At some point, these arteries just open out, and then the blood goes in between cells and returns to the atrium. The open circulatory systems vary, so in some cases there are fairly big vessels that just empty out their hemolymph, called hemolymph because it's blood-related hemo, but lymph extracellular fluid. And because this is both, it's functions like blood here, but once it gets out, of course, it's just the fluid between cells.